And so before we finish the DNA replication topic, we just need to talk about the differences between prokaryotic and eukaryotic replication. Remember that eukaryotes are complex cells which have internal organelles and endosymbiosis, including things like chloroplasts and mitochondria and things like that. And you will have also all the endomembrane system, which includes the endoplasmic reticulum, the Golgi apparatus, and nuclear envelope. And so the DNA replication in eukaryotes is going to happen inside the nucleus, and that's the cell you see on the right side. It's much more complex. It's got many linear coiled up chromosomes, and that cell will definitely replicate slower and with a more complex mechanism than the prokaryotic cell will, because the prokaryotic cell only has a single circular naked chromosome that barely has protein in it, and it doesn't really have the need to be uncoiled the way that the eukaryotic chromosome needs to be uncoiled, and also it doesn't have a nuclear membrane, so all the description of what we just talked about will happen straight away in the cytoplasm along with everything else that's happening in the cell, and it will happen a lot faster and less complex, with the lead of many less proteins. To speed up eukaryotic cell division, they use multiple replication bubbles, but that's not going to be necessary on the prokaryotic cells, which only has a single origin of replication. No replication bubbles, or at least they have bubbles, but not more than one. The speed, I said it was going to be faster. It's going to be in the order of 1,000 base pairs per second in prokaryotes, such as E. coli. One of the most common bacteria you see around, and the slower would be for eukaryotic cells 50 base pairs per second. So you can see the difference. It's a lot faster in your prokaryotic cells than in eukaryotic cells, and that actually helps explain why there actually are more mutations in prokaryotic cells than eukaryotic cells. You can think of it as eukaryotic cells have actually developed mechanisms which have slowed down the replication process to gain accuracy. And an example of this is the length of the Okazaki fragments. They're very short in eukaryotes. They're like 100 to 200 base pair longs, but the Okazaki fragments in prokaryotes are almost 1,000 base pair long, and all of this will lead to greater chances of mutations. So the adaptations of eukaryotes are all related to maintaining the structure of DNA for a long time, separating to linear chromosomes, coiling those chromosomes up, and copying them slower, and also making shorter fragments which are more manageable than the other one. The last thing, of course, is that telomeres will not be needed among prokaryotes, because since you're copying in a circular molecule, you're not going to have those loose ends that happen when you have the linear chromosome molecule. You're not going to have the problem that you have in the lagging strand of the linear chromosomes. And so there are no telomeres on eukaryotics, on prokaryotic cells which have the looped DNA. Only eukaryotic cells with the linear chromosomes will need that. Alright? So there will be difference in chromosomal structures, the amount of replication bubbles, the presence of telomeres, the complexity and speed of the replication process, and the length of Okazaki fragments. And these are just yet more examples of how prokaryotic cells are different from eukaryotic cells, which are more advanced. Now, prokaryotic cells are still complex, just not as complex as eukaryotic cells are. Alright? And with this, we conclude the DNA replication topic, and I hope you guys learned a lot, and I'll see you in protein synthesis. And meanwhile, don't do anything that would not make your mama proud.